There's another kind of carbohydrate addition that happens in the Golgi, and that's O-linked glycosylation. So in the ER is N-linked glycosylation. This was a sugar tree that was added to asparagines in the ER. Did I say in the ER? In the ER, N-linked glycosylation happens. In the Golgi, a different kind of sugar addition happens, and that's O-linked glycosylation. And what the O-linked glycosylation happens on serines and threonines. So in this picture right here, this is comparing N-linked glycosylation that happens in the ER. You can see it's on an asparagine and it's on the nitrogen of the asparagine. O-linked glycosylation, they show happening on a threonine, but serines can also receive them. And it's O-linked glycosylation because you might remember that threonines and serines both have a hydroxyl group, OH, and it is the oxygen on that hydroxyl group that receives the sugar right here. So this is sort of built up sugar by sugar in the Golgi. So when proteins exit the Golgi and end up maybe on the outside world, they can have really extensive sugars, either because they've received N-linked glycosylation in the ER that's been modified through the Golgi, or they have O-linked sugars that, or both, usually both, that were added de novo in the Golgi. Question here.
Why not tyrosine?
Why not tyrosines? The enzymes just don't recognize the shape of tyrosine. So that's a good question. You're remembering that tyrosines also have a hydroxyl group, but they don't receive this. It's just serines and threonines. But I understand the basis of your question is that hydroxyl group. Other questions? Okay, so again, it's not just O-linked glycosylation, and it's not just N-linked glycosylation. There's a ton of stuff that happens in the Golgi as proteins pass through, going stack by stack. And the reason that it can be so complex, proteins can be modified in such a complex way, is because there are different enzymes in the Golgi. So the cis stacks will have a different set of modifying enzymes. In this example, they have enzymes that are responsible for removal of mannose, versus the trans stack, which will have addition of galactose and addition of another carbohydrate. And that's possible, this complexity is possible because of the step-by-step modification. As proteins go cis Golgi, medial Golgi, and trans Golgi, they're going to be on assembly line of enzymes that do modifications step by step. Make sense? Okay, so let me show you the evidence that the enzymes exist in different stacks in the Golgi. This is an electron micrograph, and this is like a immunofluorescence picture, but this is an immuno-EM picture because you can't get this kind of resolution. You can't see the different stacks in light microscopy. So what was done was a cell was sectioned in very thin sections, and then each section was put on an electron grid, and then it was stained, counterstained with a stain that will stain proteins, and you can see the different stacks of the Golgi here in each of these three panels. So it's three different sections side by side, and then one section was stained with one particular enzyme, and you can see that it's in the cis Golgi, but absent from the medial Golgi or the trans Golgi. On the other hand, a different antibody against a different enzyme stains the medial stacks and does not stain the cis stacks or the trans stacks. And then the trans Golgi network has yet another set of enzymes. And again, this makes this assembly line of making very complex proteins in the secretory pathway possible, because secreted proteins, synthesized proteins, sort of hit these enzymes step by step in a very systematic way. Okay, so how do proteins get from the ER and through the Golgi? Yeah, question here.
In the last slide, Is it possible to use GFP on this, or to use GFP to analyze this?
Yeah, you could definitely use GFP. For example, you could tag one of these enzymes with GFP, express it, and you would see it in the Golgi. But what you wouldn't be able to see in light microscopy, which is generally what we use GFP for, is you don't have the resolution, you can't distinguish in space the difference between the cis Golgi stacks and the trans Golgi stacks. It's just too, it's too small. Okay, so the limit of resolution for light microscopy is about 100 nanometers, and these are less than 100 nanometers in size apart from each other. Does that make sense?
Yeah. So to use the antibody in fluorescence, do you have to lyse the cells, or is this like?
These are dead. That's
okay.
right. So an electron micrograph, these are really fixed, and then they're actually thin sections. So they're really dead.
Okay.
And then they're placed on an electron micrograph grid, which is tiny,
Okay.
and then counterstained right here. Or first they're incubated with the antibody, and then the second antibody in this case is labeled with something that's electron dense. So that's why we see it.
Thank you.
And then they're counterstained with something that will stain the proteins. And we can see here, these are probably ribosomes, or they could be glycogen right here. So you can have greater resolution. You can see tinier structures with the EM, but they're dead. Good question. Other questions? Some years we go through microscopy because it's really interesting, sort of the resolution and how the microscope works. But other years we don't. Albert has a really nice description of microscopy and the limited resolution and how these experiments work. Okay, so how do we get proteins from the cis Golgi through the medial Golgi and to the trans Golgi? We understand, you all understand, how proteins get from the ER up to the cis Golgi, and how is that? Go ahead. COP2 coats, right? COP2 coated vesicles bud from the ER, bringing these proteins, fusing with the cis Golgi network, and that's how the proteins get from there to there. You also know how proteins get from the cis Golgi network back to the ER, and how's that accomplished? COP1 coated vesicles. So how do proteins get from one stack to the other? And there are sort of two competing views of how this works. And there used to be two really strong camps in cell biology that at meetings would get up and actually yell at each other. It was really exciting. People felt very passionate about their experimental evidence that proved how proteins got from one stack to the other. So one camp said, vesicles bring everything else all around. So we know that COP2 coated vesicles bring proteins from the ER up to the cis Golgi network. And then the way that they get to different vesicles, different stacks, is that vesicles will bud from the ER, from the cis Golgi, and deliver the secretory proteins to the medial stacks. And then the medial stacks will deliver them to the trans stacks, et cetera, depending on how many stacks there are. It's just vesicle, vesicle, vesicle. And that made a lot of sense because, again, these are like ideas that are conserved across biology. We knew about all the other vesicles without doubt. So it made sense that vesicles would go from stack to stack. And does anybody recall how they might have learned how proteins go from through the Golgi? Did you learn about vesicles? Or maybe it just wasn't covered. Because there's another camp that would yell just as much. They never got to like fist fights, but these were really strong arguments. And that argument was this, which was actually based on an older model that then evidence began to come forward for. And that was, no, it's not vesicles right here. We go from the ER up to the cis Golgi network. And then we go from, the whole Golgi goes from the cis Golgi network to the medial Golgi network, the whole stack. So we get, let me start at the beginning. So COP2 coated vesicles bud from the ER, shed their coat. and then through the snares, they fuse together to make the cis-Golgi network. And then this entire cis-Golgi network matures. So it is carrying all the secretory proteins, and the whole stack becomes the medial stack. And that goes on, the whole stack then goes on carrying all those proteins. So it isn't vesicles budding off from each stack and going to the next one. Instead, the entire stack matures, and they keep on going up until they become the trans-Golgi network. You never run out of stacks because you have a continuous flow of vesicles coming from the ER to fuse together to make the cis-Golgi stack, which will then, as soon as that matures into the next one, you have another stack coming up. So this is called the cisternal maturation model that also had a lot of evidence to show that that was the case of how that worked. So does anybody see a problem with that model? Want to join one camp or the other? Go ahead.
Well, it'll just keep growing and growing.
Okay, so one problem is it keeps growing and growing, and what I left out was what happens at the trans-Golgi network. So at the trans-Golgi network, even in this model, we go back to vesicles. So in this model, we have vesicles coming anterograde, forward movement from the ER to the cis-Golgi, and then the trans-Golgi network, both models agree that vesicles bud from the trans-Golgi network to deliver to the plasma membrane. And we're going to talk more about that vesicle transport later in this class. So that was a detail I left out, you're right, that we need to solve. Is there something else we need to solve? Yeah?
How do specific enzymes, like for each stack, get inside at a certain time?
Yeah. So what this model needs to address is the experimental evidence that I showed you, that right here, that there are different enzymes in the cis-Golgi, in the medial Golgi, and the trans-Golgi. So if we have this maturation, so with the vesicles, no problem, you leave the resident enzymes behind. But here, if this whole cis-Golgi becomes a medial Golgi, wouldn't it carry with it all of the enzymes that are in the cis-Golgi that are doing their specific thing? And then when the medial Golgi becomes the trans-Golgi, wouldn't we also have all those enzymes carried with it, along with the secretory proteins? So, and I think this is why the vesicle maturation, the vesicle transport model was so popular, because it could explain this. And so, what is also part of the cisternal, did everybody see that problem? Okay, there are specific resident Golgi enzymes that need to live in their particular compartment. And so this model also has vesicles, but only retrograde vesicles. These are probably COP1 coded vesicles, we're not sure about this. But what we have are, when the cis-Golgi matures to become the medial Golgi, immediately enzymes bud off and bring back these enzymes back to the medial, and we'll bring back the cis-Golgi, resident Golgi enzymes, and we'll deliver them here. So we have the maturation and then immediately recycled back are just the resident enzymes that come back. Just like we saw that the KDEL receptor is bringing KDEL-containing proteins from the cis-Golgi back to the ER, this model invokes a similar concept where resident Golgi enzymes that belong in the cis-Golgi stack are immediately recycled back to from the medial stack once the cis-Golgi stack becomes the medial stack. Does that make sense? But in the pure cisternal maturation model, there's no vesicle transport of secretory proteins from stack to stack forward. Okay, so what happened to these two camps that are fighting with each other? And the reality, I think that most people accept, is that it's probably, as is usually the case when there are two extreme camps, the reality is somewhere in the middle. So different species might do things differently, but more likely there's evidence even in the same cell that both things are going on. So if we label do a pulse chase experiment, so you know how to do that experiment.